Imagine trying to rebuild a faulty airplane engine mid-flight, but every time you replace a turbine blade, the engine physically doubles in size.
Right, which is impossible to keep up with.
Exactly. By the time you fix one section, the rest of the machinery has completely outgrown your repairs, and the plane is, well, it's still going down.
Yeah, that logistical nightmare is basically the reality of treating infant liver disease.
It's wild.
Okay.
Welcome to this deep dive. If you've been following the frontier of gene therapy, you know we're moving past just reading the human genome.
Oh, absolutely. We are actively trying to rewrite it now.
Right. And today, we are tearing into a comprehensive new research paper that tackles a devastating and currently incurable condition called ARC syndrome.
Yeah, so ARC stands for arthrogryposis, renal dysfunction, and cholestasis.
That is quite the mouthful.
It really is. It is an autosomal recessive, multisystemic disease. And unfortunately, the prognosis is very grim.
Yeah, most diagnosed patients don't survive beyond their first year, right?
Exactly. And the root of this entire catastrophic cascade is a deficiency in just a single protein, which is called VPS33B.
Just one protein. And to understand the mechanics of this, we need to zoom way into the cellular level, specifically inside the hepatocytes, the liver cells.
Right, because if you're looking at the source material, the VPS33B protein is the absolute linchpin of something called the Chi-VI complex.
The Chi-V complex. All right, let's unpack that for everyone listening. What exactly does it do?
So the Chi-VI complex functions as the master regulator of protein trafficking in the cell.
Okay.
At a molecular level, it regulates snare-mediated membrane fusion.
So it's moving things around.
Basically, yes. Its primary job is to ensure that specific transport proteins are safely escorted from the cell's interior to the correct exterior membrane.
So if I'm visualizing this, it's kind of like a microscopic FedEx sorting facility inside the liver cells.
That is a perfectly accurate analogy, actually.
Okay, sweet. So in a healthy hepatocyte, this FedEx facility takes crucial bile transporters.
Like the proteins EBSC and MDR3.
Right, BSB and MDR3. And it routes them to the apical membrane, specifically into the bile canaliculi.
Which are the tiny channels where bile is actually secreted out of the liver.
Okay, so without a functional Chi-VI complex, you just have a massive logistical failure on your hands.
A complete breakdown.
The BSEP and MDR3 transporters are still being produced, but they never reach the canaliculi.
No, they get completely stranded in the wrong compartments of the cell.
And because those transporters aren't positioned correctly to, pump the bile out, toxic bile salts just begin accumulating inside the liver cells themselves.
Exactly. And that intracellular accumulation of hydrophobic bile acids is highly cytotoxic.
It's basically poisoning the cell from the inside.
Literally. It induces oxidative stress, massive mitochondrial damage, and rapid apoptosis, which is programmed cell death.
Wow.
Yeah, and this triggers a runaway inflammatory response leading to severe fibrosis, cirrhosis, and ultimately complete organ failure within just months.
Now, if you're looking at this mechanism, you listening to this are probably thinking what I'm thinking.
Why not just use gene therapy?
Right. I mean, we have the technology to deliver a healthy VPS33B gene.
You do.
And AAV, adeno-associated virus, is the gold standard for this, isn't it?
It is. It's safe, it's efficient, it's FDA approved for other conditions.
So why hasn't AAV worked for these infants?
Well, the answer lies in the fundamental biology of a developing organ.
Okay.
AAV vectors are incredibly efficient at entering a cell and depositing their genetic payload into the nucleus.
But there's a catch.
A huge catch. They do not integrate that DNA into the host cell's genome.
Oh, wait, so it just sits there?
Yeah, the therapeutic gene exists as an episome.
An episome.
Right, it's a separate, unattached, circular piece of DNA just sitting next to the host's chromosomes.
Okay, let me do a quick recap here for the listener so we're all on the same page.
Sure.
AAVs are like writing a temporary memo and just leaving it on the desk.
Yes.
Which means it isn't physically tethered to the DNA during mitosis.
Exactly. So when that hepatocyte divides to accommodate the rapid growth of an infant's liver, only one of the two daughter cells inherits that therapeutic episome.
And the other cell gets nothing.
Nothing at all. So within weeks, that initial therapeutic dose becomes mathematically insignificant.
Because the liver is growing so fast.
Right, the liver volume is expanding exponentially, but the number of corrected genes remains completely static.
So you are quickly left with an organ composed almost entirely of diseased cells again.
Yes, and worse, you cannot simply administer a secondary AAV dose.
Why not? Just give them more.
Right. You can't. The initial injection primes the adaptive immune system.
Oh, right, antibodies.
Exactly. Neutralizing antibodies are generated against the AAV capsid, meaning any subsequent doses are intercepted and destroyed long before they ever reach the liver.
Man, so you're permanently locked out after the first attempt.
You get one shot.
So treating a rapidly growing organ requires a payload that actually weaves itself into the host's DNA permanently.
The cure has to replicate in tandem with the cells. And this is what drove the researchers to utilize a lentiviral vector instead of AAV.
Lentivirus? Okay, how does that change the game?
Lentiviruses possess an enzyme called integrase.
Integrase sounds like it integrates things.
It does exactly what it says on the tin. Once the viral RNA is reverse transcribed into DNA inside the cell, the integrase physically splices that therapeutic sequence into the host's chromatin.
So it's a permanent edit to the hard drive.
Precisely. Every time that cell divides, the new genetic blueprint is replicated by the host's own machinery. The cure scales seamlessly with the organ.
That is brilliant. But the researchers didn't just jump straight into animal models with this, though.
No, you always start in vitro.
Right, in a petri dish.
Right.
They took human HepG2 cells, which are human liver cells, and used CRISPR-Cas9 to knock out the VPS33B gene.
Effectively creating a perfect model of ARC syndrome in a controlled environment.
This is where the experimental design gets really fascinating to me, because they didn't just test one lentiviral vector.
No, they tested two.
And the only difference between them was the promoter.
Yes, the promoter is key here.
Can you break down what a promoter actually is for anyone who might not know?
Absolutely. So a promoter is the regulatory DNA sequence located just upstream of the therapeutic gene.
Like an on switch.
Exactly like an on switch. It acts as the binding site for RNA polymerase and specific transcription factors.
So it dictates exactly when, where, and how strongly the gene is expressed.
Right. And the researchers compared two very distinct promoters.
Let's talk about the first one, LP1.
So LP1 is hepatocyte specific. It only turns on in the liver.
Because it needs specific keys to unlock it.
Yes, it requires transcription factors that are uniquely abundant only in mature liver cells.
Okay, and the second one was EF1.
Right. EF1 is a ubiquitous promoter. It is constitutively active.
Constitutively active meaning it's always on.
Always on, everywhere.
Yeah.
It drives gene expression aggressively in virtually any cell type it enters.
So in that isolated cell culture, that petri dish, both vectors look like absolute game changers, right?
Oh, they work beautifully in vitro. Both successfully restore the VPS33B protein.
The researchers actually used a marker called CEA to visualize the cell architecture.
Yes, and they watched these previously diseased cells completely rebuild their biocanalicular networks right there on the glass slide.
That is so cool. And the ubiquitous EF1 vector was especially potent, right? Like churning out massive amounts of RNA.
On paper, EF1 appeared far superior just due to its sheer high expression levels.
But curing cells in a dish is one thing.
It is a very different story in a living body.
Right, a living body is vastly more complex.
Evaluating a vector in a static two-dimensional cell culture completely masks the chaotic multivariable environment of a living organism.
So when you transition from a petri dish to an actively growing biological system, the brute force of a ubiquitous promoter like EF1,
it becomes a massive liability.
Which brings us to the in vivo safety trial. And this is the moment in the deep dive where this study takes a really sharp turn.
It really does.
Okay, so the researchers moved to neonatal mice, but they didn't use full-blown ARC syndrome mice yet.
No, they used mice that were heterozygous for the VPS33B mutation.
Okay, let's do another quick recap for the listeners. Heterozygous meaning?
Meaning they possess one wild-type functional allele and one mutated allele.
So they have one good copy, one bad copy. They don't have the fatal disease.
Right, they do not exhibit the fatal ARC phenotype. But they serve a very specific experimental purpose here.
Because VPS33B also does something else, right?
Yes. Previous oncology studies indicate that VPS33B acts as a tumor suppressor.
Oh, wow.
So by utilizing a heterozygous model, the researchers deliberately employed mice with reduced baseline levels of this tumor suppressor.
They intentionally created a sensitized background.
Exactly. It is an intentional stress test designed to amplify and reveal any underlying oncogenic or cancer-causing risks associated with these viral vectors.
So they injected the liver-specific LK1 vector into one cohort and the ubiquitous EF1 vector into another, and then just let them grow for 36 weeks. The results of that stress test are honestly jarring.
They are.
The mice treated with the liver-specific LP1 vector were totally clear, 0 tumors.
Completely safe.
But in the cohort treated with the ubiquitous EF1 vector, 50% developed liver tumors.
Yes, specifically hepatocellular carcinomas and adenomas.
50%. The so-called powerhouse vector was actively driving cancer.
It was. And the molecular mechanism behind this is something called insertional oncogenesis.
Let's unpack that. How does the cure cause the cancer?
Well, when lentiviral integrase splices the payload into the host genome, it does not target one safe specific harbor.
It just parks wherever it wants.
The integration profile is semi-random, yes. It actually favors regions of active transcription.
Okay.
To investigate why the tumor is formed, the researchers performed an integration site analysis.
Right.
They quantified the diversity of these viral insertions using something called the Shannon Equitability Index.
I want to pause on that metric for a second because I think it really visualizes the chaos of what's happening.
It's a great metric.
So for you listening, the Shannon Index measures diversity on a scale from 0 to 1.
Correct.
A score close to 1 means you have incredible entropy. Millions of cells, each with the virus inserted in a completely different random location, but all functioning normally.
And that is exactly what the researchers found in the healthy mice treated with the LP1 vector. Huge diversity.
But the EF1 tumor samples told a completely different story.
Their Shannon Equitability Index plummeted to 0.46.
Which means...
This indicates massive clonal expansion.
So one cell basically took over.
Exactly. It signifies that a single hepatocyte featuring a viral insertion at one specific deleterious genomic coordinate gained a huge proliferative advantage.
That single rogue cell just outcompeted all its neighbors.
Yes, dividing relentlessly to form a monoclonal tumor mass.
And they actually sequenced those tumor cells to find the exact coordinates of the sabotage, didn't they?
They did. They discovered the EF1 virus had integrated in very close proximity to critical regulatory genes.
Like the tox gene.
Yes, the tox gene was a recurrent integration site across the cancers.
And they also ran RNA sequencing and found that other genes were completely out of whack.
Right. Genes like SAT1, Big D1, and CEP78 were violently upregulated.
Violently upregulated.
Massive overexpression. Those specific genes are heavily implicated in cell cycle progression and metabolic reprogramming in cancer.
Here's where it gets really interesting to me, and I want to use an analogy here for the listeners.
Go for it.
The ubiquitous EF1 promoter acts basically like a massive, indiscriminate megaphone.
That's a great way to put it.
It doesn't matter what genetic neighborhood it lands in, it just blasts everything around it at maximum volume.
Because it acts as a robust transcriptional enhancer. It recruits a massive complex of activating proteins.
Right, so if it lands near a proto-oncogene, a gene that controls cell division, that strong enhancer activity bleeds over, accidentally blasts that gene's volume to max, and the cell gets trapped in a perpetual state of replication.
It forcefully overrides the cell's natural regulatory mechanisms, driving runaway transcription of neighboring oncogenes. That is insertional oncogenesis in a nutshell.
Okay, so that makes sense for EF1. But how does the liver-specific LP1 promoter avoid doing the exact same thing? It's still inserting randomly, right?
It is, but the LP1 promoter is highly tissue-specific and structurally reliant on hepatocyte nuclear factors, or HNFs.
Oh, right.
Its enhancer elements are relatively weak compared to viral promoters like EF1.
So it's a much quieter megaphone.
Exactly. Even if an LP1 vector integrates adjacent to a proto-oncogene, it lacks the raw transcriptional leverage to forcefully transactivate that neighboring chromatin.
So it operates quietly, expressing the therapeutic gene without disrupting the whole genomic ecosystem around it.
Yes, it does its job without waking up the neighbors, so to speak.
That isolated safety data just completely reshapes how we approach vector design, I feel like.
Oh, absolutely. You cannot just chase maximum expression.
Right, you can't just want the strongest possible promoter. The LP1 vector is definitively the safer architecture.
But, as we know, safety doesn't necessarily equal efficacy.
Right. A quiet promoter still has to produce enough VPS33B protein to rescue a liver that is actively destroying itself.
And this necessitated the definitive efficacy trial.
Time for the ultimate test.
The researchers transitioned to homozygous mice.
Okay, meaning animals completely lacking a functional VPS33B gene.
Perfectly mirroring the lethal phenotype of human ARC syndrome.
But getting the lentivirus into those failing cells introduces a massive immunological hurdle, doesn't it?
The huge one. The liver is heavily guarded by Kupffer cells.
Kupffer cells? What are those?
These are specialized macrophages anchored directly within the sinusoids of the liver. Think of them as a biological firewall.
Or like aggressive nightclub bouncers.
I like that. Yes, they continuously filter the blood, identifying and actively shredding foreign particulates.
Like our therapeutic lentiviral vector.
Exactly. They toss the virus out before it can even transfect the hepatocytes.
So how do you get past the bouncers?
To circumvent this immunological barrier, the researchers utilized claudronate-encapsulated liposomes.
Okay, claudronate liposomes, they administered these prior to the gene therapy.
Yes, as a pretreatment.
This is a fascinating bit of biochemical engineering. How exactly does a lipid bubble disable a macrophage without damaging the rest of the liver?
The specificity relies entirely on phagocytosis.
The act of a cell eating something.
Right. Hepatocytes, the liver cells we want to save, do not readily consume large lipid vesicles.
They ignore them.
But macrophages are obligate phagocytes. It is their job to eat things.
So the Kupffer cells readily engulf the liposomes. They take the bait.
They do. Once inside the macrophage, the liposome's lipid bilayer is degraded by lysosomal enzymes, releasing the encapsulated claudronate directly into the cytosol.
And what does the claudronate do?
Claudronate is a bisphosphonate. It acts as a toxic ATP analog, rapidly inducing mitochondrial apoptosis.
So it basically triggers the bouncer to self-destruct.
It transiently but completely depletes the Kupffer cell population in the liver.
The firewall goes offline just long enough for the therapeutic vectors to slip into the hepatocytes and do their job.
Exactly. And the numbers from the study prove it works beautifully.
Oh yeah, without the claudronate, the vector copy number.
Which is the average number of viral genomes successfully integrated per cell.
Right, without it, the VCN was a measly 0.2. But with the claudronate pretreatment, that metric surged to 5.0.
A massive jump. Securing high transduction efficiency is critical, but demonstrating phenotypic rescue in a murine model requires one more major adjustment.
Because mice aren't humans?
Right. Murine bile composition differs significantly from human bile.
How so?
Well, mice primarily synthesize muracolic acid, which is highly hydrophilic and substantially less cytotoxic than the hydrophobic bile acids predominantly found in humans.
So a mouse with ARC syndrome experiences liver damage, but the progression is inherently milder. Their bile is just naturally weaker.
Exactly.
So to simulate the aggressive, highly toxic cholestasis seen in human infants, the researchers had to artificially escalate the severity of the disease in the mice.
They had to create selective pressure.
So they placed the mice on a diet supplemented with 0.25% cholic acid.
Yes. This intentionally flooded their livers with highly toxic hydrophobic bile acids.
Mimicking the human condition.
Creating intense selective pressure, yes.
So under that extreme biochemical stress, the ultimate test, what did the data actually show?
The survival curves diverged drastically.
I love diverging curves.
The control cohort, so these are homozygous mice receiving a mock treatment on the cholic acid diet, they experienced rapid hepatic failure,
yielding a mere 33% survival rate.
Conversely, the cohort treated with the LP1 VPS vector exhibited an 80% survival rate.
80%, that is incredible. And the biochemical markers completely corroborated this rescue.
They did.
For you listening, alkaline phosphatase, or ALP, is a primary serum indicator of liver damage. In the untreated group, it registered at a staggering 7,201 units per liter.
Which is deadly.
Yeah, but in the treated cohort, it was suppressed all the way down to 778 units per liter.
That is nearly a tenfold reduction in active cellular damage.
And it wasn't just slowing the destruction, right?
No, total serum cholesterol and phospholipids, which typically spike to dangerous levels during severe cholestasis, stabilized to near wild-type healthy levels.
The metabolic machinery was totally back online.
It was, but the most compelling evidence of this rescue, at least in my opinion, was structural.
Oh, the electron microscopy?
Yes. The researchers analyzed the hepatic tissue using fluorescence-targeted electron microscopy. By targeting the CEA marker we discussed earlier, they were able to visually confirm the architectural regeneration of the bile canaliculi.
They literally saw the structures rebuilding themselves.
Yes. The complex interwoven networks necessary for bile transport, which had been completely obliterated by the disease, were definitively rebuilt in the treated livers.
That blows my mind.
It's remarkable science.
Okay, let's pull back and synthesize the scope of what we've covered today.
Good idea.
We started with a microscopic logistical failure, a defective Chivi complex causing toxic bile buildup. We examined the limitations of traditional AAV vectors, realizing that treating a rapidly expanding infant liver requires a permanent integrating lentiviral solution.
Exactly, to prevent dilution.
Then we navigated the sheer genetic peril of ubiquitous promoters, proving that tissue-specific promoters like LP1 are mandatory to avoid catastrophic insertional oncogenesis.
Avoiding the megaphone effect.
Right. And finally, by temporarily disarming the liver's macrophage firewall with collagenate, researchers achieved profound architectural and metabolic rescue in a lethal disease model.
You know, if we connect this to the bigger picture, the broader implications of this methodology extend far beyond just VPS 33B deficiency.
Really?
Oh, definitely. The conceptual framework established here, pairing a liver-directed lentivirus with a safe promoter and transient macrophage depletion, provides a highly viable therapeutic blueprint.
A blueprint for other things.
It offers a tangible path forward for a wide spectrum of early-onset aggressive metabolic liver diseases that have historically been completely refractory to AAV-mediated therapies.
That is so hopeful. And you know, before we wrap up, there is one final observation from the source material that I think really demands your attention if you're listening.
The selective pressure data.
Yes. As these treated mice were subjected to the highly toxic cholic acid diet, their hepatic function didn't merely plateau.
No. From week 4 to week 8, their liver enzyme profiles actually continued to steadily improve.
And the researchers attribute this progressive normalization to the principle of competitive advantage.
Right.
Think about the mechanics of that. The lentivirus delivery wasn't 100% efficient.
It never is.
Right, it didn't correct every single hepatocyte. But under the severe continuous stress of that toxic bile, the uncorrected cells underwent apoptosis and died off.
But the minority of cells that did receive the therapeutic gene were equipped to handle the toxicity.
Exactly. They survived, they proliferated, and they progressively repopulated the failing organ.
It is cellular Darwinism.
Yes. And this fundamentally challenges how we think about treating genetic disease.
In what way?
Well, the objective might not be about engineering a perfect vector that breaches every single cell. It might simply be about arming a strategic fraction of cells and letting cellular survival of the fittest do all the heavy lifting.
That is a fascinating way to look at it.
So if we can mathematically model and harness that kind of internal evolutionary pressure, what other complex, systemic, quote-unquote, incurable diseases could we force an organ to heal itself from?
It's a profound shift in therapeutic strategy.
It really is, and absolutely something to keep your eye on as these trials progress.
Absolutely.
Thank you for joining us on this deep dive. Keep questioning the consensus, keep exploring the data, and we'll catch you next time.